Good morning, everybody. We are having a little bit of technical issues with a slide presentation, but we're going to press on. My name is Amy. I have a kiddo. He's now a man with PKAN, and because of the amount of information that you're going to get to hear this morning, I'm not going to do formal introductions. I did find a quote that I loved that reminds me about what these researchers go through every day, that a person who's never made a mistake has never tried anything new. Research is a really slow process. Like, it's not the rabbit that ran ahead. It's a little turtle who's very methodical and slow. And just as a parent, I would love for these researchers to see your faces, if you can, just to remind them why they do this every single day. So if you want to allow your video to just show your face real quick for 5 seconds for these researchers to see why they do what they do. I think that will encourage their hearts, since we aren't together in person. And I love that y'all are all waving at them.
It's so great to see everybody.
Yeah, it is.
We get so consumed with running our presentations that we don't get to scroll across and see all you lovely people. Many of you.
I love this. So this is why y'all do what you do every day. And as a family member, I'm so deeply grateful. So I'm going to turn it over to Dr. Hayflick and let her get started.
Thanks so much, Amy. It's great to see everybody. You mentioned the rabbit and the tortoise at the front end. I have some art that is still up in my office from Angelica Kruken's 2 sons, one of whom has PKAN, and his picture, it's a drawing that each of them did as children. One of them is a tortoise climbing the rainbow, and the other is a rabbit climbing the rainbow. So that's a very, very meaningful piece of art for me. So I'm really pleased to be able to talk to you today about the work that's going on. Like Penny did in the BPAN group, I'm only going to be talking about one portion of it, and then Allison will talk, and then later in the session, Sue from our group will talk, and Penny will talk as well, because we've got a lot going on, which I'm very pleased about. So as you know, we have a big group. We're all focused on really different aspects of the NBIA disorders. Most of us do have some activities that are related to PKAN. And I just wanted to lay out the projects. This is actually not even a totally complete set of projects, but it's the things that we're mostly going to be talking about today. So I'll be talking a little bit about the CoAZ in the mouse work, what's next, what we're doing on that project. Allison will be talking about bone health, which we've come to understand is a very important aspect of care for people with PKAN and other NBIA disorders. Sue will be talking about gene therapy a little bit later in this session. And then Penny will be talking about the trial, giving you an update on the status of the trial. And also Allison Freed will be talking about the natural history studies that we're doing. So when I put these down as the set of topics we're talking about, we emphasize this, but part of what we find valuable in the structure of our group and in the way that we work is that we have really a bona fide bench to bedside and really everything in between. We take care of people with PKAN. We help others take care of people with PKAN. We study the disease in people. We study the disease in a really nice mouse model. We study the disease in cells. We kind of do everything in between. And I think although we're a small group, I think we're a mighty group. And part of what makes us mighty is that we have that whole translational pipeline populated with people. The other thing that I think really contributes to our impact is that, particularly for the people who join our group, they're getting to be connected to you as families and getting to learn about your families, either through the natural history study, through coming to the meetings when they're live or meeting you virtually. Those kinds of things really drive their kind of their connection to this work. So, in our laboratory, we have a mouse with PKAN. The mouse has a single mutation in the PANK2 gene, in the mouse PANK2 gene, and that causes, we now know, it's a very busy slide, and the only really thing that's important on the slide to know about is that it causes changes in the brain of the mouse that are very similar to those that we see in PKAN. So when you're kind of assessing the quality of your animal model, you have to ask how true is it to the disease? And what's true about this animal model with a single mutation in PANK2 is that it shows disease in the part of the brain where iron accumulates in PKAN. That's one aspect that's true. Another is that we see that iron is accumulating in the cells in that brain. And the third is that the dopamine system, the Parkinson disease related neurotransmitter, chemical neurotransmitter is not functioning properly. And so when we break the PANK2 gene in this mouse, we see disease in the same part of the brain as humans, iron problems and dopamine problems like we would see in a person with PKAN. And it's really those findings in this mouse that give us confidence in our model. It's also those findings in the mouse that are corrected when we feed the mouse CoAZ, or the chemical 4-phosphopantothene. They're kind of, we use the terms interchangeably. In the trial, it's called CoAZ because it's formulated into a sweet syrup, but the chemical is 4-phosphopantothene. That's the active chemical. And when we feed that to the mice, also in a sugary syrup, the disease in the brain changes, the iron goes back to a healthy level, and the dopamine changes are corrected. And so we have confidence in our model, and we have really compelling evidence that the changes we're seeing, the disease changes we're seeing, are corrected by the compound, by CoAZ. So we also have taken samples from the skin from a number of you, or your kids or loved ones, and we've grown them in the test tube. And we've actually done the same experiments that we did in mice. We did them in the humans, in the human cells. And we also showed that there's changes in iron in the cells as well, and in some of the other biochemistry of PKAN. And we showed that with 4-phosphopantothene, or CoAZ, dripped on the cells, that was an improvement to the health of those cells. You know, I've been using the term "we" to sort of describe these studies, but it's really all the work of Soo-Young Jeong. You'll be hearing from Sue later in talking about the gene therapy project, but I get to present her really, really beautiful work. So we know that CoAZ improves the chemistry in the mouse and in the human cells, but can it make people better? That's, of course, the question everybody here wants to know. I want to know the answer to that question too. But it's going to take us time to do the studies, to do the next clinical trial, to figure out what we're going to measure. And we can do some things in the mice in the meantime that are going to help us answer that question. So before answering it in the humans, we're going to answer it in the mice. Does it make the mice better? How do you know if the mice are better? What can we measure? So many of you know that the mouse with PKAN doesn't have dystonia. And while that is a disappointment to us, it's not at all uncommon for mouse models of neurodegenerative diseases not to have the movement problems that the humans have with that disease. But again, when I go back to that model and I show myself what the differences are that we're seeing that are so strikingly similar to the human disease, that's what convinces me that we're on the right track. So the mouse doesn't have dystonia, but what the mouse does have is it has night blindness, just like your kids or your loved ones. Most people with PKAN have night blindness or retinitis pigmentosa, also called pigmentary retinopathy. And it turns out that this is a very early change that we see in the mouse. It's present when the mice are teenagers, maybe children. We think we haven't looked earlier, but we think if we look earlier, we'll see that the changes are there. Maybe when the mice are just being weaned from their mother's milk. We're going to use the retinopathy to help us understand whether CoAZ can actually not just change the chemistry, but change the function of tissue by looking at the retinopathy. It's a very quantifiable measure. We can do all sorts of kind of fancy measurements of how mice behave when they're shown moving stripes. We can look at mouse vision and their behavior. We can look at structure of the retina, those kinds of things, and they're very powerful and quantitative. So right now I'm writing a grant to get funding to do a big study over a course of a few years to understand how this is helping or changing the mouse vision. So it goes without saying that as much as we treasure our mice, our goal in our work is not to improve the mice. Our goal is to understand if we have evidence to take this compound into humans for a trial to look at how it changes things in people. That's the goal of the project. It goes without saying, I think, but I think it's also worth saying. And so if the mouse vision improves, then we have a sort of a functional level, not just a molecule level, a chemical level, but a functional level of evidence that CoAZ is doing what we want it to do, and gives us confidence in a longer study in people with PKAN. So it's important to remember that even if CoAZ can correct the biochemistry, we need to show that it corrects function. And in a cell that has had a lot of damage, so in the retinopathy, we expect that for the mice who have very late retinopathy, we won't be able to change much. But for the mice who have early retinopathy, we hope we'll be able to certainly stop it, maybe slow it, maybe prevent, maybe rescue it, maybe improve it, sort of turn it around. So back to the list of projects, I'm going to hand things off to Allison now, who's going to talk a bit about the project we've started on bone health in PKAN.
Thank you. You might still be looking at Susan, but hearing me, at least that's what I've got going on here. Thanks everybody for turning on your video at the beginning. Honestly, that's the first time I've seen any of your faces in 3 days, and I think I got a little dopamine rush. It's really hard just talking out into the ether and not knowing who's listening. So I really appreciated that. So I've got 5-ish minutes to tell you about a discrete little project that we started working on last year. And that's looking at orthopedic complications in PKAN. So this is a joint project. It's our NBIA cure group at OHSU. We've partnered with Orthopedics at OHSU, as well as our friends with Manju's group at University College London, who are contributing a case or two. And probably most importantly, we've gotten our claws into a medical student who is hungry to participate and get his name into research, and he's doing some of the heavy lifting with this. So I've just got this one slide, orthopedic complications in PKAN. You know, sometimes it takes years in a rare disorder like this for us to appreciate there's a pattern of something going on, because we might only see it once or twice or once in the beginning. But over the course of time, we can say, oh, that looks like the thing that happened to so-and-so 10 years ago. And we've started to appreciate that there are orthopedic issues in PKAN. And we suspect that this is related to their dystonia, which is becoming more severe over time and contributing, as well as the integrity of their bones, because they also start to bear less weight over time and may have weaknesses there. So anecdotally, over the years, what we've seen are some pretty significant femur breaks. So the upper bone of the leg, the long bone, as well as hip dislocations that, you know, aren't just a simple thing you put a cast on and fix. These have been really intensive, several month-long issues where, in addition to having a significant break or dislocation, then the dystonia ramps up. And I don't have to tell you about the cycle there of, you know, the dystonia maybe makes the dislocation worse or makes the break difficult to treat for various reasons. And then you just get into this cycle. So that's what we've seen anecdotally, but we wanted to ask the question and be more deliberate about looking at it. So we do have a little baby protocol that we've put through the IRB. It's approved, we're ready to go. We've started systematically collecting medical records on about 7 people so far, including getting a lot of x-rays. And we want to really look systematically at what these orthopedic complications look like. If there are some we haven't appreciated, maybe, it's probably not just femur and hip. And ultimately, I think what we'd like to do is identify some common factors that might guide us towards potential treatment and or prevention. So what do patients have in common? Is there a role of dystonia in this? What is the bone health? Are there certain treatments or preventive measures that we could take to intervene in these types of orthopedic complications that really are intense for the patients and the families and have been quite problematic? And that's pretty much all I wanted to say about that, but I am seeing a comment in the chat I'm going to pull up here. How about knee x-rays? Sure, if people are having trouble with their knees, we would be very interested to hear about that. I will put my email in the chat, and anyone else with an orthopedic complication or something you'd like to tell me about, just please feel free to email me directly. I think unless you have something to add, Susan, I'm going to wrap that up there.
That's great, Allison. Thank you. I think we're set to turn it over to Susie if she's set to go, and if not, we can keep going.
Well, hi there. I'm going to give it a try. So I'm Susie Jackowski, and thank you very much for giving me the opportunity to tell you about our progress in developing a small molecule therapy for PKAN. Now, the preclinical work was completed last year, and I retired from St. Jude about 4 months ago. I continue as a scientific advisor with Therapeutics, and that's the company that's moving forward with our with the clinical trials. I'm going to try one more time to, and sorry for the video presentation, but I'm having a few technical difficulties, and I hope it's okay with you. Can everybody see this? I hope. Anyway, let me just go through the story and where we're at right now. So the pantothenate kinases, and I'll call them PKANs, control CoA biosynthesis. They initiate and regulate the entire pathway. And from the designation PANK2, you'll know that there are other PANKs that do the same function, and that's PANK1 and PANK3. And it's the mutations in PANK2 that cause the problem and reduce coenzyme A production. Now, coenzyme A, I'll call it CoA, is necessary for hundreds of metabolic reactions in our bodies. These reactions are necessary for our growth, for our function, to work, and to move. And it's really an important cofactor. Now, pantothenate, also known as vitamin B5 or pantothenic acid, feeds the PANKs that then convert it into CoA for all of these different functions. Today, I'll be talking about the panazine activators of the PANKs and CoA biosynthesis. So our approach to the therapy is to develop a small molecule that directly binds to the alternative PANK, which is PANK3, and stimulate its activity and thereby stimulate CoA production. And we've been working on this project for more than 10 years. And we've identified a whole series of compounds, a whole series of small molecules that we've synthesized and developed at St. Jude over this period of time. And our drug design was based on the PANK3 protein structure, and we were also focused on the principles that govern blood-brain barrier penetration, because obviously this is important in the disease. Now, blood-brain barrier penetration is dependent upon the chemical characteristics of the drug and also the lifetime of the drug in circulation. So we wanted to make sure that we also were able to measure the presence of the drug in the brain each time we tested a compound. We were also focused on having efficacy with oral administration to make it easier in the long run for patients to use this therapy. Now, there are several pantazines in the chemical series with strong PANK activation. And all of these have been tested extensively. We've developed new biochemical techniques and a new mouse model to enable this progress. This new mouse model demonstrates a definite brain CoA deficiency, as well as a movement dysfunction. And there's a recovery of these features in the mouse with treatment. Over the last few years, we've been optimizing the pantazines for human use, and we arrived at a clinical candidate, which I'll call BBP 671. And BBP stands for Bridge Biopharma, which is the parent company of CoA Therapeutics. And I want to acknowledge that during this optimization, we worked very closely with the experts at CoA Therapeutics who guided our development and progress over these years to arrive at this point. At the end of this talk, I'm going to give you some interesting new information that we found during our preclinical studies, and that is a new biomarker signature for the treatment response using MRS technology. That stands for magnetic resonance spectroscopy. Now, the PANK mutations are spread all across the protein coding sequence. And there's mutations that cause complete loss of the protein in cells. And then there are other point mutations that have a milder effect and can affect the chemical activity, which is shown in green here across the protein. That's the catalytic domain that mediates the chemical activity. And upstream of that, the part of the protein determines where the protein localizes or resides in the cell, and also has an effect on the total level of the protein that's found in the cell. And with all these wide-ranging mutations, it's not surprising that PKAN is a heterogeneous disease. And different mutations reduce the chemical activity to varying degrees. The activities that we've measured of some of the mutated proteins range from 0 to 100% of normal. And unfortunately, there's no clear natural history relationship between specific point mutations and disease progression. Now, we can classify PKAN as the more severe with earlier onset and more rapid regression, and that's associated with complete loss of the PANK2 protein. And this can be due to reduced protein that's produced in the cell, but also some of these point mutations cause the protein to fold improperly, causing it to be unstable and degrade very quickly in the cell and decrease its lifetime in the cell. And no protein, no activity. I also want to point out that the brain iron accumulation is not always consistent in PKAN cases. And that's one reason is because the iron pathology is downstream of the metabolic CoA deficiency. So is brain iron accumulation to a neurometabolic crisis or chronic disease or both? And can it be quantified? I think these are good questions that need to be answered. Now, as Susan pointed out, preclinical mouse models for PKAN, making them are a challenge. And actually, there's no true mouse model of human neurodegenerative disease, and that includes PKAN. There are several reviews that have been published in high-ranking journals discussing this topic, and it makes sense. Biological differences between human and mouse include size, obviously, lifespan. Mice only live 2 years. They have a higher metabolic rate than humans, and the brain organization and development is different in human and mice. For example, the white matter content in a human brain is 50 to 60%, depending upon the gender, whereas in mice, it's only about 12%. So the PKAN2 knockout mouse represents mild neurometabolic disease. And in the PKAN2 knockout that we've made in our lab, we've noted that there is a brain CoA deficiency, but it's during the early postnatal period. And when the mice reach adult stage, there's an adaptation or compensation by the other PKAN in the neurons in the brain. That's by PKAN1. And both PKAN1 and PKAN2 are equally important in mice. In humans, however, PKAN2 has a very important function, obviously, since we have PKAN disease. As Susan pointed out, there's no observable movement dysfunction in the PKAN2 knockout mice and no reduction in lifespan. However, they did find an elevation of iron in a localized area in the midbrain using a very sensitive type of detection, that is ICP mass spec, but is not detectable by MRI. The difference being that MRI can measure or detect iron in parts per million, whereas ICP mass spec detects it at parts per billion. So we made a new mouse model so that we could actually look for a replenishment of the CoA in a CoA deficient brain. We knocked out both PKAN1 and PKAN2 in neurons only. And we think this represents aspects of severe neurometabolic disease. There is a substantial and sustained brain CoA deficiency, and the mice have an impaired growth weight, a movement to function, and a shorter lifespan. But still, we just still don't see iron accumulation by MRI. That could be because mice are different than people, or it could also be because we did not knock out the PKANs in all the brain cell types, and glial cells should not be affected by that engineering, and they may contribute significantly to maintaining the balance in iron in the brain. So let me tell you a little bit more about this mouse. There's right after birth, we monitored the body weight to see the growth of the double knockout mice. It's shown in the black dots here. And you can see that they're very severely affected compared to the normal mouse, the normal healthy mouse shown by the green dots. And when we started therapy, indicated by the arrow, the red dots show the response of the double knockout mice, and there's a distinct increase in the growth rate, and that approaches that of the normal healthy mice. The brain CoA is substantially lower and measurably lower in these mice, in the double knockout mice compared to wild type. And with therapy, that CoA level is restored. The liver CoA, in contrast, does not show a deficiency in CoA. And that makes sense because there really aren't a lot of neurons in the liver. And the therapy still works in the liver and increases the CoA levels in liver as well. It increases the CoA levels in all parts of the body where PKANs are expressed to a significant extent. Now, this just shows the movement problem with these mice, which is very severe. In the two panels on the right-hand side, you can see the response of the mice We monitored their exploration and behavior over a 5-minute period using a video camera up on top of the small arena. And we measured how long they wandered around, or how often they moved, and what kind of distance that they traveled. And the wild type mice, you can see the pattern here. It's very all over the place. The Panazine therapy didn't change their behavior at all, but in the double knockout mice, there was definite improvement. It's not 100% back to wild type. This is a very severe deficiency in this animal, but there was significant improvement. So we use this mouse model throughout to test our compounds and we have several Panazines that are very active in this mouse model, and in all the other rigorous tests that we've used through the years. Now, most recently, while we were doing this work, we looked again at the MRI of the mice. As you know, brain iron accumulation in MRI imaging is a diagnostic tool that's very helpful for PKAN. And many of you know about the pattern that we see with imaging in a cross-section of a PKAN patient, of the brain of a PKAN patient, is shown on the right panel here, where the very dense region that I'm pointing to, if you can see my pointer, is associated with excessive iron in the basal ganglia. As we've, the pointer here points to a diagrammatic illustration of that. So using the same technology, or very similar, well, the same instrumentation and the same general procedure, one can do magnetic resonance spectroscopy rather than imaging, where we can look at the levels of the most abundant neurometabolites that are found in the brain. And those are very similar in mice and people. So what I'm showing here is an example from 3 types of mice. The normal wild type mouse on the far left, in the middle is the double knockout mouse model, and on the right is the knockout mouse model treated with BBP 671. And what we see, and these peaks indicate the quantity of these various metabolites, and I'll tell you about a few of them here. GLX represents glutamate, which is the major excitatory neurotransmitter, and its precursor glutamine. And those are combined in that peak together. N-acetyl aspartate is a very abundant neurometabolite. It's made in the mitochondria of neurons, and it's dependent on acetyl-CoA for its synthesis. In the few reports of MRS in PKAN patients, what is commonly found is a reduction in the NAA signal. Now, lactate is also shown here, and that's a product of glucose utilization. And the reduction in lactate in the double knockout mouse, along with NAA and GLIX, is an interesting signature in the mouse model. And when we treat, we can quantify these neurometabolites, and we find that when we treat the mice with BBP 671, the drop or the significant reduction in GLIX is fully restored following about 3.5 weeks of treatment. I didn't show the GABA, but that's the major inhibitory neurotransmitter, which is reduced in the double knockout mice, and that is fully restored by BBP 671 as well. Now, the NAA and the LAC are just partially recovered. And the NAA is an indicator of neuronal health, integrity, and function. Hopefully, with longer treatment, those parameters would increase more. So this is a neat way. It's non-invasive, and we can follow the preclinical response to treatment using this parameter. So just to summarize a little bit, PKAN is a heterogeneous disease, and there's wide variations in the patient characteristics. That really makes it difficult for the physicians to study. And different mutations cause different variations from mild to severe. And the mouse models can capture some, but not all relevant disease characteristics because of the inherent differences between mouse and human. Mouse models often represent only one type of PKAN mutation, whereas the spectrum of mutations among the patient population, as I told you, is very broad. But our neuronal PANK1, PANK2 double knockout represents severe disease with a demonstrated brain CoA deficiency. And BBP 671 significantly improves the CoA, the growth rate, the lifespan, the movement, and the neurometabolism in this model. So I'll just move on to my last slide to summarize where we are. As I said, the preclinical work was completed, and I'm happy to say that earlier this year, the FDA approved opening the phase 1 safety trial for BBP 671 in healthy human volunteers. And that study, that safety trial, is going on right now. And the results so far are very positive, very encouraging. We've also completed very long-term studies with animal models for safety and toxicity, and those have gone very well and gives us a very, very broad therapeutic index to work with when we move to figure out how to dosing the humans. After we complete the phase 1 study toward the end of this year and analyze all the data, we hope to start a PKAN clinical trial next year in 2022. And and the US FDA has granted BBP 671 both orphan drug designation and rare pediatric drug designation for PKAN. So things are looking good and we're moving forward. And I'm very excited about where we are with this whole project. It has been a long time. So thank you for your attention, and I'll take any questions.
Yeah, hello everyone. My name is Sue. I'm a neuroscientist and one of the members in the Hayflick Hogarth team at OHSU. Thank you organizers for organizing this meeting even during pandemic, but I also should have known that Patty wouldn't stop by anything. Still, I wish we were together. This was the meeting picture from Charleston 2 years ago. As Allison said, it was just awesome to see all of you in faces and in person. Today, I'm going to update you a little bit about the PKAN gene therapy project. And this project was started by Dr. Lauria Early in spring of 2019, when she was awarded with a generous gift by 2 patient family organizations, one in Italy and one in Germany. Unfortunately, she left academia and took a position in industry by in fall 2019. And in spring of 2020, we started discussion of transferring this grant to OHSU so we can continue the project. It was a long discussion at the beginning of pandemic between many organizations. However, by the late summer, we were awarded with the rest of the grant to OHSU so we can continue this project. So what is this project? What are we going to do? We have a normal human PANG2 gene unmutated, and we have viral vector called AAV, adeno-associated virus, as a packaging vector. And we package the healthy gene into the virus and then directly inject into the mouse brain, which is lacking mouse PANG2 protein. So the first part, L'Oreal assembled the virus containing human PANG2 gene, and then we transfected into the human cells. And we wanted to see whether this infection or transfection will actually make the normal human protein. So the upper panel showing here that in purple dots, you can see the full-length human PANG2 protein. And she also included a small tag to that protein called flag tag, shown here in green. And then these two signals were completely merged in a cell, showing that we're not detecting the endogenous human protein, but the one that we transfected with virus. So happily, it was making full-length human protein in a human cell. And in mouse, this project was mainly done by Dolly Zhen, shown here, our brilliant RA in the lab. And this is the normal PANG2 knockout mouse, which doesn't have any mouse PANG2. And this is actually without any surgery. And I wanted to show you, as we mentioned by Suzanne and Susie, that these mice do not have any motor problem or neurological phenotype. So they can actually move around just fine, like a normal wild-type mouse. And when we injected this virus into the knockout with surgery, this is actually 2 days later, they have no problem moving around. They have no problem eating or drinking. They're showing very healthy curiosity to the new environment. And there was no problem in wound healing, shown here in this picture with the arrow. The conclusion from this slide is that this surgery was very safe without any adverse effect. And by day 1 and day 2 post-operation, they were moving around just fine without any adverse effect. 5 weeks later, we sacrificed this animal and we looked at the brain area to see whether we can detect the virus-delivered PANG2 protein. So the first 3 lanes shown here are wild types without PANG2 mutation. And the last 3 lanes are here with the knockout. So the first part is the sham surgery, where we just do this exactly same operation, but not delivering the virus, but just a sorbitol solution into the mouse brain, just to see what the surgery is doing to the brain. So this is a wild type with the sham operation, 2 wild types with the virus injection. You can see a little bit of increase of protein. And this is a knockout mouse with the sham operation, no virus. There's no PANG2 band here, shown here in blank. And then I want you to focus on the last 3 lanes. When knockout mouse brain was injected with the virus carrying human PANG2 protein, we can see significant increase of the protein expression in both knockout mouse brain. One is stronger, the other is a little weaker. There is mouse-to-mouse variation. However, we can detect significant expression of human PANG2 protein after 5 weeks. And at the cellular level, here's a knockout with the sham operation on the left. These neurons do not show any PANG2 staining, but when we inject it with the virus, you can see these brown rings showing PANG2 expression from human protein carried by virus using this arrow shown here. These are the cells called choroid plexus and a ventricle. You see the no PANG2 expression in the sham operation, and you can see this strong expression of PANG2 protein in these choroid plexus cells. Therefore, we've performed until now 2 rounds of successful gene therapy injection into mouse brain, and we're currently analyzing data. Now, this was a very, very first baby step of this project. We just started it and we have many more questions to answer. So the first question is, how long will the effect will last? Will it last for 3 months, 5 weeks, 2 years? We don't know that yet. And also which cell types will retain PANK2 and continue expressing the human protein. The one advantage of using AAV was that they don't integrate these plasmid DNA into the genomic DNA, so it's safe and will not pass on to the next generation. However, when cells divide, they do lose these kind of plasmid DNA, but cell types like neurons will retain and continue expressing this PANK2, human PANK2 protein. So we wanted to check which cell types will retain the PANK2 expression for a while. And we also chose the direct brain injection method because it requires less amount of virus. It's directly injecting into the area where we see damage in the human PKAN patients, and we see the difference in the mouse brain. And also it doesn't get absorbed by liver, which is a typical problem for AAV injection. However, it's also invasive, and we wanted to also explore the option of non-invasive or less invasive systemic injection, and we want to find the right injection method. Now, the million-dollar question is, when can we go on for human trial? I wish I have an answer, really. This is a massive project, and we need to find experts in gene therapy and who can continue this project for the human trial. Thank you. The beautiful NBIA Cure team at OHSU, it's just an awesome team of clinicians, scientists, genetic counselors, and everybody with a warm heart. Laura Early started this project, and Shelley Wynn performed all the stereotactic mouse brain injection for us with this project. Thank you to the 2 patient family organizations for their generous support on this project. And also, last but not least, happy 25th. Sorry about that. Happy 25th NBIA. You guys are the best family association ever. Thank you.
Okay, so I am going to give a pretty high-level update of where we are with the COASY clinical trial. I know many of the folks that are involved in today's session, families that are involved, are participants in the trial. I know we also have families from elsewhere in the world who have not had access to this clinical trial, and I'll be giving a little bit of an update on that front as well. So this is, like all of our presentations, is going to be a bit of a share. So let me start things off, and then I will hand off to Alison Freed from our group. So it's problem earlier. Didn't want to advance. There we go. So as you've heard, PKAN is caused by a problem in this pathway, the vitamin B5 metabolism pathway, and the genetic mutation, the PANK2 mutation, causes a block in this pathway, and it causes a problem in how it advances. This is a pretty simplistic explanation of it all, but we'll stick with that today, I think. What COASY does is the idea is that it is jumping past this block and providing something that is downstream of the traffic jam, as Alison Gregory likes to call it, the traffic jam here caused by the gene. And with that addition there, the idea is that the pathway can then run normally again. And we had significant data from the mouse. Susan alluded to some of it in her presentation to support the idea that this was a rational approach, that it was a sound idea and a good way to proceed. The core questions that we are trying to answer in this study, and this is important, is we're trying to figure out whether COASY is safe for people to take. Does it cause any problems with other organs in their body? Does it cause problems? And is it well tolerated? Do they have side effects when they take it? And then as a secondary goal, we are looking very much to see whether it can change this PKAN biomarker that has been shown to be very robust in the PANK2 knockout mouse model. We have been in close collaboration with the FDA throughout this project from the time it was first conceived, and they've endorsed our approach for this study. We're also looking at natural history data, not as a primary outcome in the study, but as a secondary outcome. And so because so many of you, both in the US and beyond, have been participating in Pecan Ready, I wanted to have Allison go through just some high-level view of Pecan Ready so that you can see what's been going on in the natural history study. So I'll pass it to you, Allison.
Great. My name is Allison Freed. I'm one of the study coordinators for the NBIA readies that we have, as well as the CoAZ in Pecan trial. And I've been with Dr. Hayflick and Hogarth for 4 years now exactly. And I know that because my first weeks on the job were in Chicago at the NBIA family meeting, where I got to meet all of you, and also in Charleston. So please know you are my inspiration, all of you. So my goal in this presentation is just to introduce Pecan Ready to those of you who don't know about it, and then also to give an update to those that are already involved. Next slide. And the goals of the study are similar to what Dr. Sid had talked about yesterday, and you'll recognize these goals as sort of the bedrock for any successful therapy development and testing. We started enrolling in April of 2015 with the goal of trying to understand the natural history of Pecan, as well as find and identify disease markers. It was originally designed for a 5-year study, but because of everyone's diligent participation and data input, we have been able recently to extend the study to 10 years, which is another point that Dr. Sid mentioned, that natural history studies kind of need to go on. They need to go on for as long as you're trying things. Next slide. So looking at the milestone questionnaires that we asked questions from these 6 disease domains, which were selected both due to their clinical importance and also the impact that we felt that they had on patients and their families. Next slide. And this is just to give you an idea of what to expect if you were going to enroll in Pecan Ready. We have online surveys that are done every 6 months. It is a fully remote trial. There are no in-person visits. It was always intended to be online. We also have come to learn that it's helpful to sometimes call people and go over questions over the phone, which is something that we've added recently. And everybody enrolled is asked milestone questions about their disease progression. And then next slide is then in the developmental milestones are asked of people 10 years and under. And then the assessment questions are asked of people that are 6 years and older. And those are questions that come from validated, standardized parent or patient reported outcome measures for emotional health, behaviors, pain, anxiety, cognition, and motor functions. So who can join Pecan Ready? It's open to anyone who has been diagnosed with Pecan. We currently need to see a genetic test result or MRI and clinic results. We currently are only able to enroll those with a full understanding of English and have access to an internet and computer. Unfortunately, that excludes those who do not speak English and those with limited technology. We would love to be able to support the extension of the study materials to other languages, primarily Spanish. So if anybody has any interest in supporting that effort, we would definitely like to talk to you. And currently we have about 100 participants. We've done over 680 study visits. That roughly makes out to be 170,000 data points. And if we had all of those participants completing studies until the end of 10 years, we would have over 500,000 data points. So that's pretty good for a rare disorder. I wanted to shout out to people from the Visit 12. There's 7 of those participants who started with us 6 years ago and have completed their visits every 6 months. And also on that Visit 11, we've got another 17 people who in the next 6 months will have gotten to Visit 12. So thank you very much. I am really looking forward to the next 10 years of collecting more data. And you guys are all wonderful. Thanks so much.
So I think Allison just promised to be with us for another 10 years. Is that what you all got? Way to go, Allison. I love it. So now let's return to the trial. The natural history data is a key part of the clinical trial, although it is not the primary goal of the trial. We are going to be doing a secondary analysis on the data that's collected in the natural history study, sort of before COASY is started, after COASY is started, to see if it's feasible for us to detect a change. But the biomarker is really kind of the measure that is going to help tell us whether this compound is doing what we think it is. So just to remind you, this is where COASY fits in this broken pathway in PKAN. And these are the questions, the primary questions that we're trying to answer in the study. In addition to the PKAN-ready natural history study, this is a rare clinical trial that was designed from the get-go to be conducted entirely remotely. And while the last year has forced many clinical trials to be conducted partially or entirely remotely, where they can be, which is not very many, this one was designed that way completely by accident, to be done remotely from the very beginning. And we did this because we wanted to remove, because it is a low-risk compound by all measures that one can assess such things ahead of time. It is a burden on families to travel, and it often results in the deterioration of, particularly in classic PKAN children. They sometimes really don't do so well when they travel, and it can cause a deterioration in their disease that not only is terrible for the child and family, of course, but also might confound the results of the clinical trial. So we always intended for this study to be conducted remotely. Alison Gregory, Alison Freed, Katrina, and someone you don't see so often from the background, Praneet, who is our data manager for this study and website guru, genetic counselor, multi-talented individual. These are really the frontline people, back-end people who are really making the study run. And it turns out we started this study in December of 2019. We didn't have a pandemic going on at that time. And then not very long after at all, like just a couple of months later, coronavirus was part of our lives, and it very quickly actually shut down a huge number of clinical trials that were going on in the US. Ours continued without a blip because we had completely accidentally, but very fortuitously planned to do this entirely as a remote study. The participants are sent lab kits. Well, first of all, they're consented electronically, but using electronic consent and a telephone call. They are then sent lab kits to get screening labs done. They go through medical history details with our study coordinators. There's a whole bunch of screening activities that happen. And then once they are determined eligible for the study product is sent out to them. Our shipping days have become these mammoth events. This is Mafaz El-Khoury, who is our student worker who's helping Katrina on a shipping day. We have thousands of boxes going out. Every couple of weeks, we have lots and lots of boxes going out to you all. It's quite an endeavor. And then in the middle here are 2 other people who are sort of in the background. This is Dave Stone and Audrey from the Food Innovation Center, Oregon State University, who helped us develop our formulation, and they are key players in this along with pharmacy partners. The other end are the folks receiving this. happy, in this case, participants received their Coase bottles in the mail with instructions. We developed a training video to show people how to dose it, and everybody seems to be doing fairly well with that. They get their labs drawn at a lab local to their home. And then the samples that come back to us, the biomarker samples, are shipped back where Sue, who you just met, and Dolly received the samples in the lab, Katrina, and processed them for the biomarker analysis. So because of the study design and because of your energy, because there was a long anticipation and lead up to this study was enrolled in absolute record time. This blue line is showing a typical enrollment curve that would be projected for a study in rare diseases. And most rare disease studies actually fail because of lack of enrollment. They can't get people enrolled. We allowed ourselves 2 years to enroll our cohort of 50 odd patients. And by a fairly Herculean effect and your enthusiasm, we enrolled those folks in about 3.5 months. Everybody was enrolled. What that allowed us to do was to expand our enrollment and to extend the study duration. And that will give us more power to look at the things that we want to look at in this study. We have currently, I think it is 74 people in, we have consented 74 people to be enrolled in the study. There are currently 68 active. We still have people enrolled in the double-blind portion of the study. I think there are just 5 left in the double-blind portion. They will complete their double-blind phase this summer. After that, what's up next is a planned interim analysis of the biomarker. So we will take all of the biomarkers that we've collected from the double-blind phase and the first part of the open-label phase, and all of those will be analyzed at the same point. And we are also very much hoping with partners in other countries to expand the clinical trial, expand access or versions of the clinical trial to other countries, with first up probably being the Netherlands, then Australia and the United Kingdom. I think Australia, New Zealand, and the United Kingdom is where we hope to go next. Those decisions are based largely on funding in part, and in where the funding has been able to be secured, and also where we can work with the local regulatory authorities in those countries to be able to enroll people. So that is where I'm at. I'm going to pull out all of the amazing funding we have had. You all raised an enormous amount of money through Spoonbill Foundations, Stichting Lepelaar in the Netherlands. And we got a very generous grant from the Institute of Child Health at NIH, as well as these partners have funded this project along the way. And I know you've already seen this picture once, but I kind of love it. It was our team's Rare Disease Day photo with the zebra scarves and the rare disease hands. So thanks, everyone. I will stop here. And if anybody's got questions. Actually, are we done with the presentations, Susan? Can we, and Amy, can we go to questions now?
I think we're good. There is time for questions.
Okay.
So maybe if people want to put those in the chat.
And I think Susan's been answering a few of them, and Susie. Folks have been answering them as we go, I think. I'd love to see everybody again. I'm just going to look at pictures.
Well, let's all turn on our video so we can encourage these amazing researchers with why they do what they do every single day.
Hey, Hayden's family.
Shukri has his hand raised.
Shukri, are you able to unmute, or do we need to magically make that possible? Yeah. I don't think we have that ability. I think that's in the tech. So is tech support still here? If so, can you unmute the participants? Thank you.
Thanks, thanks, Susan. What's a wonderful set of work, just beautiful work. Really congratulations to your team and others who have presented today. I just want to highlight something about the quasi study. I exchanged some texts with Susan Hayflick about some new findings about PKAN. I think there was a report that suggested that PKAN is a 4-phosphopantothene phosphatase. And I will be presenting some evidence for that tomorrow as well. And I think Susan Hayflick indicated earlier that you were using the dose based on the mouse studies. It seems to be that if PKAN is really acting as a 4-phosphopantothene phosphatase, that maybe a maximum safety starting dose might be useful in this work and something to consider as you move forward the clinical trials.
Yeah, so I didn't talk about it here, but what we did in preparation for this study, and Susan, you should feel free to chime in here, is with guidance from the FDA, we were looking at the biomarker in peripheral blood, and finding a biomarker in peripheral blood in human PKAN was frankly a bit of a surprise because we have seen it as such a localized disease in the brain, but we see the same biomarker in the peripheral blood of mice, and we see it in we see it in the peripheral blood of humans too, which is incredibly helpful because we can't go in and look at their basal ganglia. Just for other folks on the, can't go in and take a chunk of their brain out the way we can with mice. We have a few more liberties with mice. Thank you, mice. But what we did is a first in human study in 3 children and 3 adults with PKAN. We had, using the mice, we had demonstrated a minimum effective dose in the mice that would change the biomarker in the brain. And using FDA and other algorithms to translate mouse to human dosing, we then did a first in human study where we looked at the peripheral biomarker over the course of a fairly short period of treatment and established that the dose range that we were planning to use could shift the biomarker. So that is, and because this is a, a physiologic compound, basically, it is something that we're intervening in a natural metabolic pathway. The classic approach of maximum tolerated dose might not be the best approach, although if there are phosphatases, that would be challenged, I understand. We'll know that when we get to see the larger biomarker set, but at least from the first in human study, suggested that the dose range that we were using was valid. And I would say that the study in the Netherlands will be looking at this, in the planned study in the Netherlands will be looking at dosing in a slightly different way and pushing the dose a bit higher, looking at some other measures, potentially pushing the dose higher, looking at some other measures. So we will get more data from that. The first part of the study, the double-blind portion of the study, was also dose ranging. So it was not a dose finding portion, but it was dose ranging, so that we will, when we do the analysis, have some data to look at what is the minimum effective dose to shift the biomarker. I don't know if that answers it, but Susan, did you have anything else to add?
No, actually, it's a very it's a small but very compelling data set. We'll have obviously a lot more biomarker data from the active trial now that will help us to have confidence. Yeah. Susie, there was a question from Allison. Can you comment on how, quote, success is defined in your current study of healthy people?
No adverse effects. None. with single ascending doses so far, up to very high concentrations. So that's, you know, that's what I know in healthy human volunteers.
Somebody else asked, do all of the researchers, stakeholders, Dr. Susie, OHSU, COA folks, ever have meetings of the minds to talk about their research and theories? We do. We have scientific meetings. We have an NBIA scientific meeting that sometimes is in partnership with the family meeting and sometimes is separate. We meet in other capacities. Some of us collaborate, but you know, we all, I think, communicate with each other about our research. I have a sense that you're getting at something, that you have a different question, and actually it would be helpful if you articulated, if I'm correct, if you articulated your specific question. So you know, it's natural for people. So we're all doing different experiments in our own laboratories, and we're seeing things that, you know, sometimes raise questions about other, you know, what we thought we knew about PKAN, or what we thought we knew about CoA biochemistry or cell biology or whatever. And we're learning things literally almost every week, month. We're learning things all the time. And so, and the way that we share that new knowledge is through publications. We confirm our work to our satisfaction. We submit a publication, and other scientists, before it actually gets published, before it goes into the public domain, other scientists criticize it. And only if it reaches a certain bar does it actually get published. I will say that in the last 5 to 10 years, the number of journals has exploded because they can make money. This is a money-making operation, and the quality of science in some of the journals is very, very low, very poor. So you have to be quite critical, but we read each other's papers, we review each other's papers, we share information when we think we can, an insight can be gained from somebody else. Yeah.
Susie, a question towards you regarding the BBP trial.
Yes, I believe that COA Therapeutics is planning to go internationally. And they've been talking with various groups in Europe and elsewhere, as well as conducting a trial in the US, hopefully. So yes, I believe so.
Yeah, so Ashley looks like she's on permanent mute. Sorry about that. I'm not able to, but there wasn't an underlying reason for the question. Just wondering, with science always changing, if you all shared your information with mouse models and discoveries and such. We do, but you know, through the typical avenues, I would say, through largely publication and at scientific meetings. Of course, you know, we sit with each other and hear about each other's work and talk, ask questions, answer questions, that kind of thing. I'm just trying to keep up with the chat, so I'm hoping others are doing so. Martin Van Kessel asks, I said earlier that both trials could potentially help each other in our kids in different ways. Can you tell a bit more? I think what I was talking about, if I'm remembering correctly from the PKAN family meeting, is the possibility that deferoprone, which is a very different targeted drug for sponging iron out of the brain and helping your body pee it out, that there may be actually, so the pantazines might work, and pantazines plus deferoprone is better, or coazy might work, and coazy plus pantazines might be better. And so there's this idea, because it's quite a different target. It's not focused on the CoA pathway. It's focused on sponging up the iron. That those may be complementary, and those are studies that we can consider, certainly, if that makes sense. Do labs exchange mouse models? Some labs do. Ours was developed with NIH funding not by the OHSU group, but by the UCSF group, our longtime collaborators and gene discovery partners in Jane Gitcher's lab. So NIH dollars are your tax dollars, and not only is there a requirement that we make the animals available to share, but it's the right thing to do. So the PANK2 mouse has been shared with several other laboratories, and it's available if somebody wants it. They simply need to write a note to me, and we do ask them to cover the cost of breeding and shipping and getting the animals to them, but we freely share, and I think Susie does too. Is that true? Or Susie's lab?
Oh,
did?
yes.
Yeah.
We've received your mouse model, your PanK2 knockout. We were making one up in parallel with yours, and we have our own PanK2 knockout mouse as well, and it shows exactly the same features that you described. And we have shared our mice with a variety of labs for different reasons. Freely. And that is a policy of St. Jude Children's Research Hospital, that we're very open and transparent about all of the work that we do and try to make things publicly available.
Penny, there's a question maybe for you. Do you expect the CoAZ dose to go up? You're on mute there, Penny.
I was typing, and so I didn't want to be thundering away in everybody's ear. Yeah, Gaetano, not within the trial as it is currently designed. So it is possible we would test other doses in the future. As I mentioned, the trial in the Netherlands is designed to look at some of those. It's designed to look at that in a slightly different way, in a much more limited number of people. And so if that were to inform, together with our data, were to inform dosing, we would consider what the next best steps are at that point.
Yeah, so Gaetano asks if we've tried the compounds on each other's mice. We're very interested in testing Chukri's compounds in the PANK2 knockout mouse in Sue's model. And we've shown in the published paper that we tried Pantothenate, we tried CoA, and we tried Pantothenate in our mouse, and none of those worked. None of them did what 4-phosphopantothenate does. But I don't know, Susie, have you looked at any of the other compounds in your animal models?
We've looked at Pantothenate by itself. We've looked at Pantothenate. We tried to reproduce that study, and we weren't able to see any effect. And we've tried acetylphosphopantothenate, and there's no effect there.
Yeah, so it's again, it's important to remember that the mice they're different models. Yeah. Other questions? Other ones I missed?
It looks like we have somebody who's raising their hand, Mr. Mamon, if you want to ask your question.
Yes, thank you again. To Dr. Hayflick, are you guys considering new delivery mechanisms? There are some serious efforts. I know at Yale, working with people in bioengineering departments, especially working on nanoparticles, and they have developed some new nanoparticles that can deliver drug small molecules, can deliver metabolites as well as viruses and guide RNAs directly to the brain after systemic infection. We'd be very happy to introduce you to them. We're actually working with them on some of these applications. This will be something to consider as well. Maybe that's what maybe limits the efficacy of some of the treatments that are applied today. But I think direct delivery to the brain or even to specific areas in the brain, in this case, might actually be helpful. Just a comment.
Thank you. Yeah, that's great to know. There's actually some work going on at OHSU too for resolving eye disease using nanoparticles, and also for lung disease and cystic fibrosis. So that's great to know. We actually, I have an interest in nanoparticles for delivering the PKAN protein, because how else are you going to reach all of the cells in the brain? That's the challenge with PKAN. In PKAN, because my view is that the disease sits very locally in a small region, and we have shown in our mouse that by injecting the virus with the human gene into the mouse, the same part of the mouse brain, we actually can see after the mice are sacrificed and we cut their brain, we can see that the virus is actually reaching the target area in a way that's, and we can use much less virus. So there's a lot that's appealing about that in PKAN, but you know, we never put all of our eggs in one basket and put our blinders on to other activities that should be going on in parallel. And I think nanoparticle delivery it's an emerging area. So thanks for that comment.
Yeah.
Susan, can you share our Facebook support group with everyone? It's support for parents of children with PKAN. We would welcome them into the group as long as they answer the security questions. If you can't, I understand. I think I can. I think I just did. That's from Janice Heasley, Aaron. Yeah, like Penny said at the front end, it's really wonderful to see your families here. I'm looking at Max and hey, Max, good to see you, dude. Yeah. Yeah. Hi. Hi. The Hups are here. The Nitopes are here. The Bonfilios, the Leaps. I'm not going to call you out because I'll forget somebody,
Hey,
but my heart is warmed. Oh, but I do have to call out the Carters there. Hey, Melissa. Hey, Brian.
Ooh.
High fives to you guys. Yeah.
Susan, Ashley Miller has a hand raised.
Okay, go for it, Ashley. Can you, there you
I'm
go.
on mute now, yay.
Hey.
I don't have my camera on because I am logging on in between housework and yard work, and it's like 90 degrees here, and I'm just a disaster. So I did want to ask the mouse model comparisons. I might be saying all of these wrong. I'm not a scientist. I don't know what you guys do. But the OHSU PKAN mouse model only has the brain biomarkers, correct? I might be misspeaking, but you all had mentioned that there's no physical movement dysfunction with the mouse model for your PANK2 mouse, but it sounds like Susie's mouse model, or one of them, I don't know if it's the new mouse model that she made or their PANK2 or PANK2 or what, however you say it, does have physical movement dysfunction. Does it have, is it missing the brain part of that? Is it, what is the difference between these mice?
Good question.
Okay, I'll answer that. No, the mouse model with the movement dysfunction is different from the original PANK2 knockout. And that's because we knocked out two genes, both PANK1 and PANK2, and we did it specifically in neurons, not in the entire mouse, because we wanted to generate a severe or a substantial CoA reduction that was sustainable, that we could test with therapy, with our proposed therapy. So they're distinctly different. And the newer mouse model that we made as has a whole range of problems that are due to the CoA deficiency in the neurons. I'm sorry for the confusion about the mouse models. We did make a PANK2 knockout in our lab that matches very closely the behavior of what Dr. Hayflick's mouse has, okay? And it really doesn't have a movement dysfunction and so on and so forth. So we wanted to generate a new model so that we could really test the therapy.
Forgive me
so-
if this is ignorant.
So can I
but-
just make one more, just add one more point to that? So the PKAN mouse model at OHSU, the one that Sue works on, is it has, as I said, it has these biochemical changes in brain. It does have a very robust retinopathy, vision problems, and this is really quite closely in parallel with what we see in people with PKAN. In my mind, the value of the PANK2 only knockout, as opposed to the St. Jude's double knockout, the value of the PANK2 only knockout is that my best scientific opinion about what's causing PKAN, low CoA, something else, doesn't come into play, right? I have to trust the model to tell me what PKAN is. And the St. Jude's model is based on good science, but a presumption that low CoA, it's a disease model that is intended to have low CoA, and then to use their compounds to drive up CoA. Ours is less assuming that we know that there is low CoA. I think that's an important distinction.
CoA production is really the only known function for the pantothenate kinase, and there's no other one that's been demonstrated or shown in years since CoA was discovered.
So there's a paper that's
pancakes.
percolating. Yeah, there is a paper that's percolating that suggests alternate activities for pantothenate kinase that isn't yet, it's not yet ready for publication, but so there may be more, I mean, there may be more to learn.
That isn't to say that it doesn't have other functions as a sensor in these different locations in the cell. It actually senses whether to turn on CoA synthesis or not, depending upon the metabolic context that it's in. And all the PANKs are located in different parts of the cell where they act as kind of as thermostats, multiple thermostats, to maintain the homeostasis, the CoA supply that's necessary for the function of all these different parts of the cell. So that could be what you may refer to as alternate functions, but I'd be interesting to know the data, to see the data on this percolating paper.
I am too.
I think that we are out of time and we want to honor schedule. I am thankful for all of y'all's formalized curiosity, researchers, and that you're poking and you're prying, that it has such a purpose. And so we are thankful for you, and we will close this session for right now. So thank y'all.
Love to everybody.
Thanks,
Thank
everybody.
you.
Oh.
Okay, good to see you.
Thanks for your attention and interest.
See you at the dance party. Adam to phone. I want to see your tattoo there, dude. We're showing our tattoos at the dance party, Adam. Okay? Get going. See you guys.